All right, welcome back, fellow scientists. So this is what my voice sounds like on Tuesday. It's actually getting better, so hopefully I will be back with you on Wednesday. Um, yeah, at the earliest, Friday at the latest. Anyways, I'm not gonna let a little thing like a head cold, laryngitis, whatever you want to call it, keep you guys from learning. So we're gonna press on ahead. We're still, if you remember our learning objectives, I don't have my smartboard software on my home computer, so I apologize, but go back in your notes. You remember our learning objectives, we were starting with history, right? And then we're solving genetic word problems. You just took a quiz on that, so hopefully that went well. We're still kind of looking a little bit at the history. We're going to summarize Mendel's contributions, and then we're still kind of in the category of solving genetic words problems. But guess what? As amazing as Mendel was, we've learned more since the 1800s, surprisingly. So there were some things that we now know about genetics that Mendel didn't know. So this PowerPoint, we're going to start looking at those. So you already have most of this written down, but just kind of follow along in your notes and just make sure that you actually have everything. So we already talked about how Mendel determined there were chemical factors, right? He didn't call them genes, but we call them genes, passed on from generation to generation. So you already have that written down. We already talked about the different forms of the gene that he called alleles. So you already have that written down. And then we already talked about, excuse me, how each adult is diploid, right? And then when they make gametes through the process of meiosis, how each gamete is haploid. And we talked a little bit about with his experiment with the green and the wrinkled and the round and the yellow, how the dominant characteristics don't always stay together and how the recessive characteristics don't always stay together. So I think this one is new. So go ahead and write that down. Ask the sub to pause if you need more time. And then this is the big one, is that alleles for different genes usually assort or segregate independently from each other. So we could have a yellow wrinkled pea, or we could have a green round pea. If you'll remember, the round and the yellow characteristics were dominant, and the green and the wrinkled characteristics were recessive. And they don't always have to stay together. They can assort, they can segregate, they can separate from each other. So go ahead and write that down as well if you don't have that written down already. All right, so now, like I said, Mendel knew a lot. He figured out a lot, but he didn't know everything. We've actually discovered some things since Mendel. So if you'll remember all the way back to his first initial observation, is that a red flower mated with a white flower made a pink flower. Like, what happened to that observation? Is that still true, or was he just like, was it an optical illusion? Well, it was still true, right? It didn't happen in pea plants, but it happened in other kind of plants, and we now call those types of alleles, or those genes, we call them incompletely dominant. So go ahead and write that down. That's one of our key terms or key phrases that we need to know. And this is a case where neither allele is completely dominant. So it didn't happen in pea plants, but it does happen in 4 o'clock flowers. So we're going to kind of model Mendel's experiment, right? And we're going to take a true breeding, a homozygous red 4 o'clock flower, and mate it with a true breeding homozygous white 4 o'clock flower. And the result is, no kidding, a pink or a heterozygous flower. So I don't think I need to say it, but I'll say it anyways. Write all of that down. Ask the sub to pause because I'm going pretty fast. The free software that I'm using only allows me to record a 15-minute video. So I'm going to go fast. Ask the sub to pause if you need more time. So now we have this problem though, right? We've used to use the capital T for the dominant allele and the lowercase t for the recessive allele, right? But if neither allele is completely dominant, like, what do we do, right? I'm still of the opinion that you still need to use the same letter, right? If you remember, I don't think I called this out explicitly, but if we're looking at the trait of plant tallness or plant height, right? We didn't use a T and an S for tall and short. We used a big T, little t, right? So if we're talking about one trait, we need to use one letter, right? So how in the world do we do that? There are different ways to get around that. I like to do it this way. I like to use a base letter. So I'm using C because we're talking about flower color. And then I like to use a superscript to say what the allele actually is. So C to the R, right, is the red allele. That's how I kind of signify or notify that. And then C to the W is the white allele, right? If you want to use R and W, Like, that's totally fine. If you want to use a big R and little r, you can do that. That's totally fine. But on quizzes and on tests, excuse me, and in our notes, this is how I'm going to represent it. So if you want to use that, great. If you have a system that works better for you, that's great. I won't stop you from using that. So this looks like true breeding or homozygous red, and then true breeding homozygous white mates, and then 100% of the F1 generation is pink, or C to the R, C to the W, and here we have our pink 4 o'clock flowers. All right, so that's incomplete dominance. Now, another non-Mendelian trait, or beyond Mendel as I call it, is called codominance, right? Before, it was almost like neither allele wanted to take charge, and so they just kind of melded. They just kind of blended, right? That's not true with codominance. With codominance, both alleles are dominant. So we see this in chicken feathers. So we're going to take a true breeding homozygous chicken with black feathers, right? True breeding homozygous chicken with white feathers. And if this were incomplete dominance, what would we get? Yeah, that's right. We'd get a gray chicken. But because it's not, because it's codominance, we get a speckled chicken, or a chicken with a mixture of both black and white feathers. So again, I don't have to say it, but go ahead and write it down. Right? You should have written down the Punnett square from the last example. If you didn't, then look on with someone who did and copy that down. So again, I'm using my base letter F because we're talking about feather color. F to the B is a black feather, and then F to the W is a white feather. So then we make the Punnett square. Again, go ahead, write this down in your notes. And then we get, there is our speckled chicken right there. So our F1 generation is 100% speckled chicken. All right, so those are two beyond Mendel traits. We have incomplete dominance and we have codominance. When I come back on block day, then we'll have a worksheet and we'll have practice problems with this and everything like that. But right now, I'm just trying to get through content. So third non-Mendelian trait, or beyond Mendel, are called sex-linked traits. Now, to understand this, we have to have a little bit of background. So humans have 23 pairs of chromosomes, and one pair of chromosomes, the 23rd pair of chromosomes, are called the sex chromosomes, right? So humans have the sex chromosomes that we call X and Y. And if you have two X chromosomes, then you are a female. And if you have one X chromosome and then a Y chromosome, then you are a male. And here we have our picture of our X chromosome over here and our Y chromosome over here. Now, what do you guys notice? Yes, that's right. The X chromosome is a lot bigger. The Y chromosome is a lot smaller. I like to call the X chromosome the encyclopedia, and I like to call the Y chromosome the comic book. Now, if you want to take from that females are more complicated and smarter and have more information and maybe like to talk more, you can do that. I'm not saying that, but if you want to imply that from biology, if you want to take that males are kind of like I don't know, simpler, and we don't have a lot of language and we don't have a lot of information, right? You can do that. I'm not saying that. Right, but if you want to make that, if you want to draw that conclusion, you can. Now, because the X chromosome is so much bigger, by the way, write all of this down in your notes. I don't think I have to say that, but I'm going to. So, because the X chromosome is so much bigger, there are some genes that are only on the X chromosome that are not on the Y chromosome, right? There's lots of information that's in an encyclopedia that's unfortunately not in a comic book as great as comic books are, right? So a couple examples of that are genes that cause color blindness, which we're going to use that as our example going forward in the PowerPoint. Genes that cause hemophilia. So hemophilia is the inability to clot when you get a cut, is the inability to stop bleeding. And then muscular dystrophy. So that's a problem with muscles, where as a person gets older, then the muscles get weaker and weaker and weaker. So there's a particular protein that controls muscle development that's found on the X chromosome, not on the Y chromosome. So like I said, we're going to use color blindness as our example. So just really quickly, just a little bit of background, right? What does it mean to be colorblind? Well, here's a classic colorblind test. So if you're not colorblind, you should be able to see that this says 25, this says 29, this says 45, 56, this says 6, and this says 8, right? Now, if you are colorblind, your eyes, your retina, doesn't make the proper cones to be able to distinguish, excuse me, primarily between green and red. So, for instance, while you can see 25, you can't see 29, right? This is, even if you're color seeing, this is made to look like you're non-color seeing, like you're colorblind, right? You can't see the 45, you can see the 56, you can't really see the 6, and you can't really see the 8 if you were colorblind. So these circles over here, right, look like these circles over here to people who are colorblind. All right, so being colorblind is recessive, right, to seeing normal colors. You didn't have to write anything down for the last slide, but go ahead and write this down. So again, here we need to keep track of two things. We need to keep track of our chromosome, right, and then we need to keep track of the allele that controls cone formation and contributes to color blindness or non-color blindness. So go ahead and write this down. X to the big B, X to the big B is a female who's color seeing. X to the big B, X to the little b is a female who's color seeing because, again, it's recessive, so she has one dominant allele, but she is a carrier. And then X to the little b, X to the little b is a female who's colorblind. And then on the male side of the house, we have X to the big B, Y, is a male who's color seeing. Now help me out. Why doesn't the Y have a B associated with it? That's right, because males are comic books. We're simple. We don't have a lot of information, so we don't have that gene that makes that cone, right? We just have one gene. So if this gene is messed up, if this allele is messed up, if it's mutated, if it's the recessive allele, then we don't have a backup copy like females do, right? So then X to the little by is a male who is colorblind. So it only takes one copy of a recessive allele. In males, it only takes one copy of a recessive allele to be colorblind. So that's why males are more often colorblind than females. So to do Punnett squares for colorblindness, right, we have to do, first of all, a Punnett square for gender, right? So females, right, they're usually listed above the box, and so they just have an X chromosome, so they can only contribute an X chromosome to their offspring. Males have an X and Y chromosome, so males can contribute either an X chromosome or a Y chromosome. So you'll notice that according to this Punnett square, 50% of babies born are female and 50% of babies born are male. That pretty much lines up with what we see. Now, here we go. I'd like you to do this before I show you the answer. So work with a friend, work with a partner. The sub is going to pause the video, right? A normal non-carrier woman marries a colorblind man. What phenotypes and genotypes will we see? So pause the video, go ahead and make that Punnett square, and then come back and I'll show you the answer. All right, so here's the answer. We have our normal female, we have our colorblind male, and then those are the genotypes that we would see. For phenotypes, right, this would be a color-seeing female, this would be a color-seeing female, this would be a color-seeing male, and this would be a color-seeing male, right? So that's why colorblindness, you may have heard like some traits skip a generation. So that's one reason why we can say that they skip a generation, because even though dad is colorblind, it hides in the female because dad only gives his X chromosome to his daughters, to his female offspring, and they have a backup copy. They have a big B from mom. His sons he gives a Y to, right? But that doesn't have the information, so they just rely on their backup copy, or their only copy rather, from mom. All right. So now, if one of the daughters from the last problem, right? So you got to remember what that was: X to the big B, X to the little b, marries a color-seeing man. What genotypes and phenotypes will we see? Again, go ahead and pause the video, do that Punnett square in your notes, and then we'll come back and we'll look at that. So here we go. Here's the female from the last problem, right? Marries a color-seeing man, X to the big B, Y, right? So there's our genotypes that we're going to see. Here is a color-seeing female. Here is a color-seeing female who's a carrier. Here is a color-seeing male. But then here in our F2 generation, right now we have that trait show up again of a colorblind male. All right, well, that's almost 15 minutes, and so unfortunately I am out of time. Hopefully this was enough material for you guys I should be back, like I said, on block day or at the very